Welcome back. Plant anatomy looks at the internal tissue structure of roots, stems, and leaves, explaining how plants grow, transport water and nutrients, and support their own weight. In this video, we'll cover meristematic and permanent tissues, the internal structure of roots and stems, and secondary growth. Let's begin. Plant growth is localized to specific regions called meristems, made up of meristematic tissue, cells that are thin-walled, have dense cytoplasm, lack large vacuoles, and remain capable of active, continuous division. Meristems are classified by location. Apical meristems sit at the very tips of roots and shoots, driving growth in length, called primary growth. Intercalary meristems occur at the base of leaves or internodes, commonly seen in grasses, also contributing to primary growth. Lateral meristems, including the vascular cambium and cork cambium, run along the sides of roots and stems, and they drive growth in girth, or thickness, called secondary growth. All the other tissues in a mature plant, which have stopped actively dividing and instead perform specialized functions, are called permanent tissues, and they're derived from these meristems through a process or differentiation. Permanent tissues are divided into simple tissues, made of just one cell type, and complex tissues, made of more than one cell type working together. The three simple tissues are parenchyma, thin-walled cells that provide storage and, in green tissue, photosynthesis, kalenkyma, cells with unevenly thickened walls that provide flexible mechanical support common in young stems, and sclerenchyma, cells with thick lignified walls that are dead at maturity, providing rigid, mechanical strength. The two complex tissues are xylem and phloem, together forming the plant's vascular tissue. Xylem transports water and dissolved minerals upward from roots, using dead, hollow conducting cells called tracheids and vessels, along with supporting fibers and living parenchyma. Phloem transports the products of photosynthesis, mainly sugars, both upward and downward, using living conducting cells called sieve tube elements, which remarkably lack a nucleus at maturity, assisted by companion cells that manage their metabolic activity. Cutting across a young dicot root from outside in, you first encounter the epidermis, a single protective layer with root hairs for water absorption. Beneath it lies the cortex, several layers of parenchyma cells for storage. The innermost layer of the cortex is the endodermis, a critically important single layer of cells, with a band of waxy suberin deposited on their radial and transverse walls, called the Casparian strip. This strip forces all water and minerals to pass directly through the endodermal cell membranes, rather than slipping between cells, giving the plant selective control over exactly what enters the vascular tissue. Just inside the endodermis lies the pericycle, from which lateral roots originate, and at the very center lies the vascular tissue itself, with xylem, typically arranged in a star-shaped pattern, and phloem located between the arms of that star. While roots and stems initially grow only in length through primary growth, dicot stems and roots, unlike most monocots, also undergo secondary growth, increasing in girth over years, which is exactly how trees form wood and grow thicker trunks over time. This is driven by the vascular cambium, a lateral meristem that lies between the primary xylem and primary phloem, and it produces new secondary xylem toward the inside and new secondary phloem toward the outside each growing season. Because secondary xylem production usually exceeds secondary phloem production and happens at different rates in spring versus autumn, this seasonal variation creates visible concentric growth rings, commonly called tree rings, which can be counted to estimate a tree's age. A second lateral meristem, the cork cambium, forms nearer the outer surface and produces the protective outer bark, replacing the epidermis, which cannot stretch to accommodate the increasing girth and eventually gets shed. Let's recap. Meristematic tissue drives all plant growth, apical and intercalary meristems for primary growth in length, lateral meristems for secondary growth in girth. Permanent tissues split into simple types, parenchyma, kalenkyma, and sclerenchyma, and complex vascular tissues, xylem for water transport and phloem for sugar transport. The root's endodermis, with its Casparian strip, tightly controls what enters the vascular tissue, and secondary growth, driven by the vascular cambium, produces the wood and bark of trees, with the vascular cambium making new xylem and phloem, and the cork cambium making protective bark. That's anatomy of flowering plants. Being able to sketch a root and stem cross-section from memory will serve you well on exam day. See you in the next video.